Hi, everyone. It's really great to be here, and thanks a lot for the invitation. So to continue the exotic animal models and to go even one step further, I will be talking about reptiles today. And before I start, I want to show you one slide with motivation, why we want to study these animals in general, or have a large diversity of model systems. So this is an image of a chair. This is an abstract chair, and it is not like any other real chair that you may find out there. Yet it captures the fundamental principles that characterize the chair, so some anchoring to the ground and the place to sit on. And one of the ways by which we can extract these generalizations or these principles is by looking at many different chairs. And by examining the commonalities between them, we can understand what are the principles, and by looking at the differences, we can understand the specializations. So for example, this chair has wheels, which hints on its special function. Now, when we come to study complex biological phenomena, we can basically use the same type of approach and utilize the huge diversity of model systems, of animal model systems that we have on Earth, and basically do the same trick. Now, this is one reason why we want to have large diversity of model systems. The second reason is the Croc Principle, according to which, for many questions in biology, you can find the optimal model system to answer this question. And indeed, if we look at the history of neuroscience, we have many of these examples, and I show some of them here, and maybe the famous ones are the axon of the squid, studied by Hodgkin and Huxley, or the neuromuscular junction of the frog, studied by Bernard Katz. Now, why can we do that? Why can we take these axons of the squid, who diverged from us about 650 million years ago, and say something about the axons in our brain? And the answer for this is, of course, evolution. So this is the evolutionary timeline. And Earth was created about 4.5 billion years ago. First vertebrates appeared only around 500 million years ago. And basically, there are a lot of constraints. On the one hand, the physical world gives us, and that leads to the same solutions to problems in the world that animals face. And of course, we have inheritance. So these are the two reasons. And today I want to focus on the period that starts here in evolution, so about 400 million years ago. And what is interesting about this period is that life begins to emerge out of water, and animals start conquering land. And if you think about these animals, they had to face a lot of new challenges. So movement is much more accurate on land, for example. And yesterday we heard from Kate about the influence of gravity. So gravity starts to take place, is important when you move to land. And in general, navigation is different. And sensory modalities change, and they can do better in some cases. And just to give one example of vision, so these are two images, one underwater and one taken on land. And because light attenuates very strongly in water, you can basically see for very short distances. Whereas in land, you can see large distances, and there's an increase of 1 million-fold in the volume that you can perceive on land. And indeed, if you look at evolution and you track the sizes of the eye sockets, you see that upon this transition, there's an increase in the size of these eye sockets to take advantage of this property. Now, one thing that you can do when you have very good vision is to start planning. Okay? So if I'm an animal here and I want to reach this point, and there's some predator lurking here, and I can see around, so I can choose the better ways or the better routes to reach this safe location. And I will get back to this point of planning later on. So what happens in the brain of these animals? Okay? Presumably, these animals had to adapt to these changes, and these adaptations, we should see them in the brain. So this is the cladogram of vertebrate of evolution. And we see three main animal classes. So here we see the split from tetrapods to amphibians, and here later the split to reptiles and mammals. And if we look at the brains of these animals, so this is the frontal cross-section through a brain of a mouse, a turtle in this case, and an axolotl here, we can see that there are marked changes in the pallium of the brain, so in the forebrain and specifically in the shell of the brain. So the general organization actually is conserved. So on the one hand side of the pallium, we'll have the medial pallium, which in mammals is the hippocampus. And there's evidence that it's responsible for memory in all of these animal classes. On the other side, we have the lateral pallium, which is the piriform cortex in mammals and responsible for olfaction. But chunked between these two, there's the dorsal pallium, or the neocortex in mammals. And this neocortex is, in fact, not a mammalian invention. And it actually appeared first here at this point in evolution, about 320 million years ago, in the common ancestor of mammals and reptiles. And if we look at this brain area in reptiles, we can see that it's already structured. Okay? It's a structured and layered area. It does not have 6 layers as in mammals. It has only 3 layers, and it's smaller. But the basic organization principles are already there. Whereas if we go to amphibians, there's nothing there yet. But actually, interestingly, what you see here specifically is a stain for SADB2, which is a transcription factor, and is used as a marker for cortex. And you can see how it nicely expresses here in the neocortex and here in the dorsal pallium of reptiles. But also in amphibians, there's a small area here, a group of cells that begin to express this marker, but it's still not organized. So this is very interesting to us, but I will not talk about this today. And this is here to remind me that there's another brain area that is added in this stage in evolution. This is the dorsal ventricular ridge, this large area under the cortex, and people are still debating on the homology of this area. And I'll get to that during my talk. So the first thing we wanted to look at was sleep. And why sleep? Because sleep in many ways is simpler than looking at awake animals. So there's no sensory input arriving to the brain. And so we wanted to characterize the sleep patterns. And we chose these animals, which are live in Australia, bearded dragons. And this is because these animals are really, really good sleepers. Okay? So unlike rats and mice that sleep transiently and are nocturnal, spend 15 minutes sleeping, then wake up, these animals can appreciate a good long night's sleep. So what you see here quantified is the head angle as a function of time. The gray box is the lights off period. And you can see that as we turn off the lights, they lay their head down, they close their eyes, their heart rate decreases, and they stay like this for a couple of hours until they wake up in the morning. So really, really good sleepers. Now what about brain activity? So in mammals, we know that during sleep, we have 2 brain states. We have a REM sleep state, which is characterized by low amplitudes and high frequencies. And we have the slow wave sleep state, which is characterized by high amplitudes and low frequencies. And we have transitions between these states with a cycle of about 1.5 hours in humans, 30 minutes in cats, less in rodents. Although we don't really know why this specific organization of neural activity is useful for. Now, what about other animals? So almost, all mammals basically have some kind of 2-state sleep. Later, it was found that also birds have the same 2 states of sleep. But what about reptiles and maybe amphibians? So here, the situation is not clear. People tried to ask this question in the past, but all of it was with very old techniques and recordings from outside of the brain. And there were also contradictory results. So eventually, the field converged on the notion that these animals do not have 2-state sleep, and that's it. So we wanted to revisit this question using modern techniques. And so we implanted linear probes in the brain of these lizards. Now, this is not so trivial because in reptiles, the brain moves more than in mammals. And so part of our goal is to develop probes that move with the brain. So these are probes we developed which are 4 microns thick and move with the brain. But this is not enough. We're currently working with Yael Hanin from Tel Aviv University to develop even better probes. But we can use them to record LFP and spiking activity and start asking these questions. So this is how activity from the brain will look like. This is raw data, 1 electrode. And this is during sleep. Just by looking at it, you see that there's something very clear and very organized going on here. There's this clear oscillation. And if we zoom into a small section, you can see it's composed of 2 phases. One with high amplitudes and one with low amplitudes. So of course, we want to look at the spectral profiles. And so you chunk the data to small pieces, calculate correlations of the spectral profile, and reorder them, cluster them, in this case with a dendrogram, and you find 2 main classes. One of them, which appears during the high amplitude signals, has relatively low frequencies. And the other, appearing in the low amplitude signals, has relatively high frequencies. So they're really reminding us what we know from mammals. And this is just a quantification over animals and days to show you we see the same thing. We can take this activity and to quantify it and track it throughout the night, we take the ratio of the delta to beta band. The details are not important, but this allows us to really track this oscillation. So this is how it looks like. What you see here is this delta to beta band color-coded. So blue is one phase, and yellow is the other phase. And this is 30 minutes of recording, and then we put the consecutive 30 minutes of recording one below the other to represent the entire duration of the night. Okay, so here we turn off the lights, and you can see that as we turn off the lights, the brain begins to oscillate really robustly between these 2 patterns. This oscillation continues for a few hours, and then it begins to decay in amplitude. Eventually, we turn on the light, the animal wakes up and continues to move. Now, this is super robust. This oscillation, we see it for different animals, different nights, really repeating itself. This really clear and robust oscillation. And it's so oscillatory that some people who looked at our work thought that maybe we were measuring artifacts of some unknown source. So, but how do you address this thing of an unknown artifact? So what we ended up doing to really prove that this is real is taking 2 lizards, putting them one beside the other. And so if there's some external unknown artifact, we'll see the same oscillation in the 2 lizards. But in fact, what we saw is that each of them had its own oscillation, and they were not locked. So they're real, and we have other evidence for that as well. The cycle is very fast. So this is the autocorrelation function. It's about 80 seconds. I remind you, in humans, it's 1.5 hours. It's temperature dependent, which is really interesting because the whole system scales up and down with temperature. So we usually work with 27 degrees, where it's 80 seconds. But if we cool the room to 24 degrees, it's increased to 110 seconds. And if we heat the room, it decreases to 60 seconds. So somehow the whole neural circuitry organizes with temperature and changes its rate. This oscillation, the period increases slightly over the night. Now, what about eye movements? Right, this is the hallmark of a REM sleep. So to look at that, we basically use infrared cameras and track the animal and the eye throughout the night. And we use an optic flow algorithm. So basically, each vector here represents a movement of groups of pixels, and you can sum them to get this green trace. And in blue, you see this delta to beta oscillation I showed you before. And every time we have this peak here, it means that the eye made a small jitter. And already here, you can see that the peaks sit on specific places in this oscillation. And to really quantify this, you take now all these oscillation cycles throughout the night. So we have 300 of them or so. And this is what you see here. So each row is one cycle. The color code is for this delta to beta ratio. And the red dots are these eye jitters. And you can clearly see that these jitters fall on one of the phases of sleep, specifically the one that is similar to REM sleep. And again, this is some quantification over animals and nights, showing the same type of phenomena. So taken together, both in terms of amplitudes of the signal, frequencies, and eye movements, we have something that is very similar to what we know from mammals, mammalian sleep. Now, what are these sleep patterns good for? Right, so there are many theories, and maybe the most dominant one is that during sleep we consolidate memories. So we acquire memories when we are awake. We acquire them in the hippocampus for temporal storage, and then we move them during the night or later to long-term storage in the cortex. And I should mention that this process is thought to be mediated by a few activity patterns, namely the slow waves that we see in cortex, and I showed you before, and the sharp wave ripples in the hippocampus, which are these potential deflections with a ripple with a high-frequency signature sitting on top of them. Now, if we go back to the data I showed you before and zoom in to the slow wave sleep section, you can see that it's actually made of a series of ripples. And zooming in and filtering, you can see that each of these sharp waves has a ripple on top of it. Okay? And we have thousands of these ripples occurring every night. Now, what I did not tell you yet is where we record it from. And here you see a frontal cross-section of the brain. And when we actually traced the position of the electrodes, we saw that they are partially in cortex and partially in this DVR area, this huge area that sits under the cortex. And actually, all these oscillations that I described before were not coming from cortex or from the hippocampus, but actually from this dorsal ventricular ridge. So we wanted to know what is the interaction between this area and the cortex. So for that, we go to spiking activity. And here you see a typical raster plot. So lines are spikes of neurons. In purple, neurons in cortex. In red, neurons in DVR. And here you see the LFP. And we have these sharp waves here. So we take all the sharp waves, we collect them, and look at the activity of a single unit surrounding the time of these sharp waves. So this is what you see here. Here are 5,000 sharp waves, 1 unit, and it's firing in all these sharp waves, and the sharp wave is at time 0. And you can see that for a unit in DVR, it's really locked to the sharp wave. So it fires in the falling phase of the sharp wave. And if you do it for many units, you see the same. So this is the firing rate. The line is the time of the sharp wave. Basically, you see that they are all locked to these sharp waves in DVR. What happens in cortex? So in cortex, actually, it seems like not much from here. But here you can see already there's a slight increase in firing rate after the sharp wave. And if you look at the averages, there's a clear gradual increase of the firing rate after the sharp wave. And you can see it here again overlaid. So this is the locking of neurons in DVR. And this is this delayed increase in activity in the cortex. Now, this really reminded us of what people have shown for a long time ago for the interaction between the hippocampus and the prefrontal cortex. So in the hippocampus, during these sharp wave events that occur during sleep and underlie this memory consolidation phenomena, we have an increasing firing rate in the hippocampus. And then in prefrontal cortex, we have this delayed and slower increase in firing rate. Now, in mammals, we know what happens during these sharp waves and what is consolidated, right? We know that when an animal moves through a linear track, and we heard about it yesterday, it has in the hippocampus place cells that will fire in some sequence as the animal moves through the track. So each neuron will be responsible for a specific position along the track. And then when the animal moves, we have this sequence of firings. And now we know that during sleep and during sharp wave, we have basically a repetition of these sequences. So they are condensed in time, but basically the same sequences are repeated again and again. But actually, we know today that it doesn't happen only during animal movements. So we have these sharp waves also when the animal is silent. So when it finished going through the track and it sits in a state of quiet wakefulness here, there are also sharp waves. And if we look at the sequences, we find that these are the same sequences. So they can be in the reverse order or in the forward order, but we have a repetition of the same type of sequences. And furthermore, if we look before the animal even started moving, so at this point, we also see repetitions of these sequences. And this is thought to be related to planning, so that an animal, when it reaches some junction, let's say, and he has to decide if to go right or left, it has this replay of these sharp waves with different possible trajectories. So these events may be related to planning. Now, going back to the lizards, I showed you these sharp waves during sleep. But actually, also when you go and look at awake lizards, and this is a preliminary data, I should say, you find the same sharp waves. In fact, you find the same two states of sleep. So this state and this state. And in mammals, we know today that there are also states during wakefulness, but the differences are much more subtle. Here, we see very strong differences between states during wakefulness. But in contrast to sleep, where the transition between these states is really structured with this 80-second cycle, here it's all mixed and probably depends on behavior. And this is what we are trying to find out now. So how are these sharp waves related to the behavior of the animal, and how does these states change with behavior? And so we adopt some technologies that were developed by Arne Meyer to track the eye movements. And so we're trying to see now where the animal looks, what are the visual stimuli it receives, and how this relates to these sharp waves that we see. And this is just to show you that these are cool animals and do cool behaviors. So this is a bearded dragon playing ant crusher. So you can see that, first of all, it's very good at it. Yeah, it's better than most people I know. And also notice how it looks at the operator for reward, maybe. No, not yet. So these are very nice animal models, but there are not enough studies of behavior to really allow us to ask these questions in a very quantitative way. And so this is one of our main challenges. Okay, so just to conclude this part, so what I've shown you is that we find clear evidence of two states during sleep of bearded dragons, of a specific lizard, which makes it more plausible to us that the common ancestor of birds, reptiles, and mammals already had some basic signature of two states of sleep. But what we see is that this transition of states is really an oscillatory process and looks much simpler. And if I were to study the transition between these states, this would be a very good model system because we have hundreds of these transitions every night. Another thing we are interested in, I mentioned, is the patterns of memory transfer. So while in mammals it's mainly studied the interaction between hippocampus and cortex, maybe this is a generic thing. And so if you want to move information from one brain area to another brain area, and in our case we see this between the DVR and the cortex, maybe you need a specific type of neural organization. So this is something we want to understand. And the fact that we have similar states, or at least that's what we see now, similar states between the sleep and the wakefulness, maybe hints that at the beginning there were two main activity states that were different in wakefulness and sleep, and maybe they had common functions, and we are looking at that. So now in the time that I have left, I want to switch to a completely different project. It's a bit more methodological, and it's in these animals, these turtles, red-eared sliders, which come from North America and Mexico, and we grow them here in the zoological gardens of Tel Aviv University. You can see a few of them basking here in the sun. And what is special about these animals is that during winter, they hibernate underwater, okay? But they hibernate for 3 months underwater, okay? So to do that, they had to develop mechanisms that allow them to deal with this lack of oxygen. And I'll show you how we utilize these mechanisms. But before that, I want to just introduce the visual system of these animals. So we are interested in vision, and so the visual information comes to the eye. From the eye, it goes basically to 2 places. One of them is the optic tectum. We heard about it just a minute ago in fish. But the other pathway is that they go to the lateral geniculate of the thalamus, and from the lateral geniculate, the information goes up to the cortex, okay, through the lateral forebrain bundle, and you can see here in the cross section. So basically, it comes here and arrives into the cortex. So this is a circuit that we find also in mammals, and in the cortex, we have this structure that I mentioned, where we have basically 3 cell layers. This is the main cell layer with excitatory cells. Here, basically the information comes here to the upper layer 1, where it impinges on the apical dendrites of these neurons, and the axons go out here to layer 3. And another thing we have in layer 3 is this population of inhibitory neurons that sits really here in layer 3. Now, all of this structure sits above a ventricle, which is nice because now what you can do is you can take an electrode array, and without penetrating the tissue, you can just stick it on the tissue here from the ventral side and measure neural activity, because these cells are really close to the ventricle. So this is what we do. And now coming back to the point of the anoxia resistance, what we can do is do all of this process ex vivo. So not in the behaving animal, but rather in the dish. And this is, again, unique to reptiles because of this anoxia resistance. You can take the brain out of the animal, put it in a dish, not cut it. And we don't do slices, we do whole brain thing. And it continues to be active for a few days. And during this time, you can, for example, keep the eye intact, with the optic nerve connected, and project images to the retina and measure the responses in cortex. But now you're working in vitro, so you have all the advantages of in vitro work. You have eye accessibility, you can combine multiple techniques easily, you have stability, the brain does not move, everything is there. And we have sensory responses in this brain, because we can stimulate it and see how the neurons react. And for that, as I said, we use these multi-electrode arrays. We can record LFPs, and we can record spiking activity. And because we see the spikes on many different channels, we can also do very good spike sorting and separate single units. And eventually, we end up with rasters like that, where these are cells, this is time, and here you see responses to static images that we show to the retina. And of course, we're interested in these activity patterns, which I'm not going to talk about today because we actually don't know yet what exactly they represent. So this is something we're working on. I will talk about a different side project that came out of this. And this answers the question of how much you can push extracellular electrophysiology. So usually we have traces like that we can record from an electrode, and we do 2 things with these traces. We either filter it in the low pass band, and then get the LFP signals, or we high-pass filter the traces and get spike sequences. But now we have this huge growth in the number of electrodes that we put in the brain. And if you can imagine a situation where we have many, many electrodes in every position, and these electrodes, they measure basically currents, right? Because that's what they measure. They measure transmembrane currents. And these currents are everywhere, right? In synapses, in channels. So if you have a dense enough distribution of electrodes, maybe you can get information about all of these currents and not just LFP and spike sequences. So this is the mission. And what we did here is something very, very simple, okay? So after sorting the spikes very well, we look at the row potential without filtering surrounding the time of the spike. This is what you see here in gray. So all the traces are just the activity surrounding the spike, and you can see it's very noisy. I should mention that we have to choose these traces in a certain way, so we choose them when the neural activity in general is not very high. I'll explain later why. But when you average these traces, you can see the spike here, and then after the spike, you can see this slow potential following the spike that lasts really several tens of milliseconds. Okay, and we were wondering what these potentials are. Of course, we can record them from many electrodes because we have many of them. And we can represent them like this as a color code. So each dot here is one electrode, and the color represents the peak here. And if you start by classifying them and just you look at the flavors of these signals, you find two flavors. In one case, there's the spike, and then the signal goes down. In the other case, the signal goes up. And looking at large population, we see these main two clusters. So immediately you think about excitatory and inhibitory neurons, right? So to look at that, what we do, we take this brain on the multi-electrodes array that sits here. And now we cut the rest of the brain. We just end up with the cortex. The cortex is still intact. Okay, it's not sliced, but only the cortex. We can patch a single cell now. And I should mention that it's silent, right, because there's no input arriving. So we patch a single cell. We can inject current, induce it to spike, and we can also see the spikes on the MIA. Now, when we patch the cell, we know a lot of things about the cell. So we can characterize the dendritic structure, if it has spines, doesn't have spines, other electrophysiological properties. And so we can know if it's an inhibitory or excitatory neuron. And indeed, what we find is that for inhibitory neurons, these currents go up after the spikes. And for excitatory neurons, they go down after the spikes. And to verify these are really synaptic currents, we just use synaptic blockers. So this is before. And when we wash in the blockers, the spike is still there, but the slow currents are not there anymore. And this is specific to the blocker. So excitatory blockers will block the excitatory currents, but not the inhibitory currents, and vice versa. So these are really synaptic currents. So what we think is going on is that we have a neuron. It has an axon, and it has terminals, and we have these electrodes sitting next to it. And when it fires, the action potential propagates, and then we have all these post-synaptic currents in the synapses. And we basically can measure them with these electrodes. But this is really great because we can take a data set that before we only took two kind of types of information out of it, and extract another type of information, which is something about the structure of the axons. You usually have to do stainings, and here we can also connect directly the activity of the neuron that we record during the experiment with the structure of the neuron. Now, to prove that, what we need to do now is to really verify this correlation between morphology and activity. So to do that, we actually have to register these two data sets. This is not trivial, but we found a way to do it with tracking blood vessels. And this is the result. So in color code, the lines are the axons of a single neuron. The color code is for the depth. So the red are deep axons that are close to the electrodes. And the blue are superficial axons far from the electrode. And in the background, you see basically the recording from the multi-electrode arrays. This is not the LFP, but rather the current source density, okay, which is just a transformation of the LFP to get to currents from potentials. And you will see what happens now following a spike. Okay, so this is the spike, and then after the spike, we have this slow potential. Okay, and if you notice the positions of these currents, you see that they are correlated with the position of these red axons. So the axons that are close to the multi-electrode array. So here and here. So there's really a correlation between these two parameters. And we can take out of it a vector, okay? So we triangulate the position of the cells from the high frequency signature of the spike, and then take a vector to the center of this field. And I'll get back to these vectors in a second. Now, another thing we did is we put everything to a model to see that everything fits. So again, we can use volume conductance theory to estimate, given the morphology of the neuron and some assumptions, what would be the potential that we measure from the electrodes, and then compare it to the potential that we measure. And you can see that there's a very good fit, which really convinces us that what we're seeing is basically a signature of the axon of this neuron, or actually the functional part of the axon, so the postsynaptic currents of the neuron. And in any given experiment, we have hundreds of these, right? Because we record many neurons together. So we can get this information from many neurons in each experiment and do statistics. And these are the statistics. So what you see here, this is the distribution of these projection vectors on the tangential plane of the cortex. Okay, so each dot is a neuron in one experiment. And this is the distribution of the angles for inhibitory neurons. So for inhibitory neurons, you see that there's nothing much going on. So different neurons project to different positions in cortex. But when you look at excitatory neurons, there's a clear bias. And the bias is to the lateral directions, which means that neurons preferentially project from the medial part of the cortex to the lateral part of the cortex. And now you can take many experiments, and basically you see the same thing. So this is just an average over all these points for many, many experiments, and you can see the same bias that I showed you. And then we went back to the handful of reconstructed cells that we had, and actually also there we find a slight bias in the same direction. But finding this bias from this sample is very hard. First of all, each neuron like this to reconstruct it is days of work. And here we get information from hundreds of neurons for free, basically. And also the statistics are very hard, yeah, because again, you have a handful of neurons. So we see this bias, but why is it interesting? What can it tell us about the function of this circuit? So this is a top view of half of the brain of the turtle. This is the olfactory bulb. This is the cortex. This is the area that we record from with the MIA. And the black is a stain for myelin. So these are these axons that come from the lateral forebrain bundle and innervate the cortex. And something that we know from other studies already is that whenever you project an image to the retina, there's a wave that propagates through the brain. Okay? So and you can see this wave here by the cross-correlation function, or here just by looking at the average LFP over stimulations. There's really a wave that propagates, and it propagates from the lateral part of the brain to the medial part of the brain. And this happens regardless of the visual stimulation. Now, we don't know if there's a functional role of these waves, and in general of waves in cortex. So what we think is going on, and I remind you that we found that the excitatory cells project to the other direction. Okay? So if you remember, this layer is rich with inhibitory cells. And this is basically the area that is close to the electrode. So this is where we find our fields that we measure. And we find that for this population of cells that project here, there's a bias to this direction, so to the lateral direction, right? But here, these cells activate inhibitory neurons, which do not have a bias. So the inhibitory neurons inhibit this area. And so if the input, the thalamic input, comes here and begins to propagate, then this mechanism basically does not allow it to go back. And this can serve as a mechanism to propagate these waves forward in one direction. So this is just an example of what you can do with this type of information. And since I see that I have to finish soon, I'll skip this part and summarize. So what I hope I have convinced you is that we have a very nice model system where we have basically a system that is intact, it's sensory responsive, but we have all the advantages of working in vitro, and this could be very useful for a specific type of questions. And I showed you data from arrays of 256 electrodes and 120 electrodes. Today we have these CMOS arrays with 26,000 electrodes. And we can do all of these with very high resolution. And using this stability, we were able to show that you can extract these projection fields from single neurons and say something interesting about the dynamics or the mechanisms that allow specific dynamics in these cortices. And with that, let me just thank the people involved. So most of this work was done in the lab of Gilles Laurent during my postdoc. The lizard work was done with Janie, Sam, and Huapeng. The turtle work with Mike and Lawrence, and these are the technical assistants. And for a few months now, we're building our lab. This is how it looks like, or at least it looked like about 3-4 months ago. And hopefully in a month, it will look like this. So, and we have open positions for students, and so you're welcome to contact me. And thank you for your attention.